Hmm?
Ma'am, this is my first class.
This is your first class?
Yes, ma'am.
Okay, just 1 second, dear. Just 1 second. All right, so you're in class 12th?
Yes, ma'am.
All right, okay. Please switch on your video. Let's start today's class.
Yes, ma'am.
Also, what all chapters have been completed at your school? Am I audible, Inaya?
Yes, ma'am.
All right, yeah. So I was asking what all chapters have been completed at your school?
Actually, we are in the inheritance chapter.
Okay, you are currently doing the inheritance chapter.
Yes, ma'am.
Okay, and where in the inheritance part?
The dihybrid cross we just finished. We started the codominance part.
Okay, so how are you liking class 12th? Is it better than 11th or
Yes,
worse?
it's better.
It's better?
Yes,
11th was gruesome, isn't it?
yes, ma'am.
Okay, maximum students have this reaction to 11th only. Now you might be finding 12th interesting also and easy also, right?
Yes, ma'am.
Hmm. All right, so yeah, in my batch also we're doing the same chapter, but the introductory part. Okay, and
Okay.
how do you find this particular chapter, the principle of inheritance and variation? Is it easy?
Uh, yes, till where we have done now, because it's the same part from 10th, it's easier.
Oh yeah, yes,
Yes,
yes.
it's
It
the
resembles a lot
repetitions
of,
and
yes, it resembles a
yes,
lot of 10th. Yeah, but with its own depth and everything.
yes, ma'am.
Oh, So let's start with this chapter only. And what about the first three chapters? How are they? The sexual reproduction in flowering plants, human reproduction, reproductive health. How
Uh,
did
the
it
f-
go? Hmm.
yeah, they were also good. The first two chapters are done, and the test is also done for us. And the third chapter,
Okay.
this test is still pending.
Okay. And do you have any upcoming tests in this month?
Not in this month. From June 3rd, we have our unit test.
June 3rd, you have your unit test. And
No,
have you
no,
gotten
wait,
your
June 6th, we have our unit test. Yeah,
June 6th?
yeah.
From June 6th, or do you know the date for biology?
No, wait, I don't know the date for biology, but it's from 6 to 13.
Okay. June 6th onwards. And do you know the portion?
For biology, it's the first two chapters: reproduction in flowering plants and animals.
Oh, great. All right. Okay, so now let's talk about principles of inheritance and variation. Now, this is a bigger branch of science that we are entering into. This branch of science is the genetics, right?
Yes.
Now, we are stepping into the world of genetics and we're going to go knee deep. 10th was only superficial knowledge. Now, we're going to go knee deep into the genetics part. Now, we are going in this particular chapter we're going to talk about what are the principles as far as the inheritance and the variations are concerned, what principles govern these two things. But before learning about the principles governing them, we must know what inheritance and variations are separately, right? So
Yes.
talking about the inheritance, now inheritance, it is the process by which the characters or traits are passed from parents to the offspring or to the progeny. We know that, you might resemble either your father or your mother, isn't it?
Yes.
It happens for everyone. Like I resemble my father, and then my father resembles my grandmother a lot. So you see how the thing comes being transferred. Like my grandmother is like a different generation, then comes my father's generation, and then it's my generation, right? So
Yes.
from generation to generation, certain traits or certain characters, certain similarities, they are being passed on. And this whole entire phenomena by which they are being passed on is known as inheritance. We call this method of passing on the characters, traits, any quality from one generation to another, mostly from the parents to the offspring, is inheritance. Here,
Yes.
all right. Now, since this process of inheritance is being governed by sexual reproduction only, isn't it? Wherein the two gamete formation takes place and then those gametes they fuse in order to form the zygote. Now, we have studied that sexual reproduction is equal to variation. Now, what is variation? It is the introduction of some new characters into the offspring, right? Something that is different from the parents. So variation, it is the degree by which the progeny differs from the parents. Some quality or some trait, like, okay, like I may resemble my father, but I am not his exact replica, right? I have my own features, I have my own qualities. But then if someone were to see us side by side, they would like, okay, they are father and daughter, but I to some extent differ from him also. So that is variation. That extent is variation. So the degree by which the progeny differs from the parents is variation. Or the other way to define variation is that it is the introduction of new characters in the offspring. So when new characters are introduced in the offspring, that is variation. And do you have any idea what variation leads to? Variation leads
Uh,
to dash. You know this?
Evolution.
Evolution, correct.
Yes.
So variations, they lead to evolution process. Now, coming and talking about genetics, so it is that branch of a science that deals with the study of inheritance and variation. This is a different branch of science all in on its own. Just like how we have botany, that is plant science, we have zoology, that is animal science, then this is the science that deals with the concept and how inheritance happens, how variations happen. And if we say genetics, Mendel comes along with it. Like we can't mention genetics without the mentioning of Mendel because he is the father of genetics. He was the first person to ever talk about it, talk about how the characters are being transferred, and he was the first person to lay out the ground rules to explain as to what things are happening and how they are happening. Clear?
Yes.
All right. Now, why he is known as the father of genetics is because he was the first to demonstrate the scientific basis of inheritance and variations by his hybridization experiment. So, by the use of his hybridization experiment. Now, when you were studying sexual reproduction in flowering plants, you might have heard about hybridization,
Yes.
right? Wherein what we do is that we take two superior parents, like we take qualities from two superior parents, and then we want to combine it into one offspring. So that for example, there is this parent A. Parent A contains good qualities such as abiotic resistance, meaning it can withstand any drought, it can withstand any flood or anything. It has that quality, but it does not have the quality to grow anywhere.
Mm.
The B parent, like the next parent that we're taking, it has the quality to grow everywhere. So we take these two superior plants, and when they are made to fertilize together, then the offspring that is formed contains the better quality of both the parents. This is hybridization, isn't it? Now,
Yes.
the same thing he did, but for his experiment, he chose the garden pea plant, whose scientific name is Pisum sativum. So he chose the Pisum sativum plant for his hybridization experiment. Now, while you're writing Pisum sativum anywhere in your answer, please pay attention to the scientific rules that are followed for nomenclature, right? The
Yes.
genus name must start with a capital letter. The species name must start with a small letter, and they must be underlined separately. So this rule, it is 100% must to be followed because there are many teachers that cut marks on this. You know, once you've like not written it nicely, there are many teachers like That cut marks on this. Okay. So
Yes, ma'am.
now, why did he choose pea plant? Why not any other plant? Why not rose? Why not hibiscus? Why not cucumber or any other plant? Why did he choose only pea plant? To be fair or to answer this question in very easy way, the first thing is that, Mendel and his family was like their family was farmers.
Hmm.
So they had a big tract of land. Tea was easily available to him. So the first reason he chose tea is because it was easily available to him. Right? He did not go out
Yes,
of
ma'am.
his way to check it. So pea plant was near his house only. But coming on to the biology aspect of why did he choose the pea plant? So the first reason, the first and the most important reason is that pea plant has many contrasting characters or many contrasting traits. And all in all, it has 7 pairs of contrasting traits. Okay?
Yes, ma'am.
Yes. Now it produces a large number of seeds and completes its life cycle in one season. Flowers show self-pollination, meaning flowers show autogamy. Okay, do one thing, don't note down anything right now. First you understand, I'll give you time to note down or take down the notes. Okay.
Okay.
Also, yeah, one more thing I forgot to mention. If while I'm teaching, if there is anything that bothers you, like maybe you did not understand anything, maybe there is a doubt, you can stop me in the middle. You don't have to feel shy or hesitant doing that. Okay.
Yes, ma'am.
Also, is my pace of teaching fine for you?
Yes, yes, that's fine.
Okay. All right. So the first reason, the most important reason, the pea plant has many contrasting traits, 7 pairs of contrasting traits. Then in one life cycle, it produces a large number of seeds. So just like how you might have done titration in
Hmm.
chemistry, right?
Yes, ma'am.
Now, while you were doing titration, your teacher might have said that take many readings, take as many readings as possible. Normally, when we conduct any experiment, we are advised to take many readings because we should have many observations so as to decrease the scope of error. Same way here, if there are many seeds, it will be advantages only because there will be less scope for error and there will be more observation to look at. So that is why pea plant was very beneficial because it produced a large number of seeds. And then it completes its life cycle in one season, meaning after flowering, after producing seed, it dies. So it has a very shorter lifespan, one cycle. Now, the flowers of the pea plant, they show self-pollination, that is autogamy, and it is true breeding, meaning if any plant shows self-pollination, it means that it is true breeding. So if it shows self-pollination, then we do not have to take the pollen and transfer it. We do not have to do all of those things, isn't it? So it
Yes, ma'am.
shows self-pollination. Apart from that, since it shows self-pollination, now if we want to introduce any external pollen, so we can cut the anther by emasculation, right?
Yes.
And we can introduce the new pollen. So that is why it is easy to artificially cross-pollinate them. The hybrids, also another reason is that after cross-pollinating, hybrids are produced and these hybrids are fertile, meaning these hybrids, when they will be used to reproduce further, they will do so because they are
Hmm.
fertile. So these are the very many reasons because of which Mendel chose the pea plant. Clear?
Yes, ma'am.
Any doubts?
No, ma'am.
Okay. Now let us talk about the 7 pairs of
Oh.
contrasting characters or the contrasting traits that are there in the pea plant. The first one is seed shape. Now seed shape is round or it can be wrinkled.
Ring.
The seed color, it can be yellow or it can be green. The flower color can be violet or white. The pod shape may be full or constricted. The pod color may be green or yellow. The flower position can be axial or terminal. Axial is when we draw like the normal way, this one, when the flower is this type in position. Then this is axial. But when the stem terminates into one flower, that is terminal position. So the flower can be axial or it can be terminal.
Okay.
And then the stem height. The height of the plant can be tall or the height of the plant can be dwarf. So these are the 7 pairs of contrasting characters. Another thing to remember that one column is for the dominant characters. One column is for the recessive ones. Now, dominant characters are the ones that will show themselves in most of the time. Recessive characters will get masked by the dominant character. So, for example, if a dominant character is present and then a recessive character is present, the dominant will be shown, the recessive will get masked. Okay, we'll talk
Yes.
more about it later on. But for now, is everything clear?
Yes, ma'am.
All right. Okay. Just give me 1 second. Just 1 second. Okay?
Yes.
All right. Now let us look at some of the important terms. Do you people have any doubts up till now?
Mm.
Okay. Marzia, are you there in the class? Can you hear me? Marzia, are you able
Yes,
to?
ma'am.
Okay. You weren't there in the class yesterday?
Actually, I forgot about the class.
Oh, you forgot about the class. Okay. Anyway, this is what I've taught in the class only. Okay. Also, will you
Okay.
be able to switch on your video?
No, no.
Why? See, the only reason why I ask you people to switch on your video is because I want to see you. I want to have like an interactive session. Because in that way, you will understand better, and even I will feel nice that, okay, I'm not talking to myself, I'm talking to someone because I have a face to see. That is why I ask you to turn on your video. Anyway, moving on to the important terms. Now, the first and the most important term is the gene. Now, gene is the functional unit of inheritance, meaning it is the part which is responsible for inheritance that happens. Like inheritance happens because of genes only. Now, Mendel, when he discovered genes or when he thought that there was something being transferred from one generation to another generation, so he called it as factors at that time. He called it as factors. He did not know that it was genes, so he gave it its own name by calling them as factors. Now, the next thing that you must know about is alleles. Now, the genes which code for a pair of contrasting traits. For example, see here. For example, seed shape. Now, the seed shape is round also, it is wrinkled also. So, round and wrinkled are the alleles of each other because they are opposite of each other. Round is a full shape like this, and wrinkled is like this. So they are complete opposite of each other. So we can call them as alleles of each other. So the genes which code for a pair of contrasting traits are known as alleles. For example, if capital T is used for tall and small t is used for dwarf. So both the capital T and the small t are the alleles of each other. Okay. Any doubts in this thing?
No, no.
Okay. And one more thing. Let me tell you, how do we choose what alphabet? Now see, in this chapter, you might have noticed that there are many crosses. You have to make crosses in order to find out what is the gene and how the gene orientation is. Now, for making crosses, you can select alphabets. For example, if you are talking about the height, so height is tall and dwarf. So for example, if you are taking capital T for tall, so you must take small t for dwarf. Okay. Small t for dwarf and capital T for tall. Because both the alphabets should be same. You can also take capital D for tall and small d for dwarf. But remember that both the alphabets that you're taking, it must be same alphabet. Only capital and small can be changed. Okay. So that, you know why? So that it does not make any confusion for you while making the crosses. That is. Now, after that, we'll talk about characters. The character is the feature of the individual. For example, the stem height. See, okay, first let me explain traits. And traits are the distinguishable feature of the character, such as tall and dwarf. So here, if we look at the chart, so if the seed shape is the character, it is the feature of the plant, and seed shape being round or it being wrinkled. Round and wrinkled are the traits of the character seed shape. Same goes for everyone. Also remember one more thing, that these 7 pairs of contrasting character, these are the ones that were considered by Mendel. No other character can be put here. Okay. So while your sometimes happens is that we get long answer question, and maybe you forgot one of them, one of the characters. Now you would think that, okay, I will imagine some character and write down. That will make your answer completely wrong, because these are the 7 pairs that are the exact 7 pairs which Mendel took. So you can't change it. Okay. So learn it just as it is. So characters and traits you people have understood. Any doubts in character and traits?
No.
All right. Now let's talk about homozygous condition and heterozygous condition. Now, the true breeding characters having 2 identical alleles, for example, capital T when present together is homozygous condition. Homo means same. Homo means same. So when capital T are present, we'll call it as homozygous dominant, because both of them are dominant. And when small t are present together, we'll call it homozygous recessive. So homo means same, and in this condition, the 2 genes, or you can say the 2 alleles, they are similar, they are identical. Then comes the heterozygous condition. Now hetero means different. Now here, one capital T, one small t, and this is the case of a hybrid. Okay. So in this case, what happens is that one is dominant and one is recessive. So 2 different alleles of a particular gene are present. And remember, always remember this thing, that homozygous condition is of 2 types: homozygous dominant, homozygous recessive. Any doubts up until now? All right. Now let's talk about the next term, that is genotype. Now genotype refers to the genetic makeup of an organism, whereas phenotype is the externally observable character. Now, if capital T is coding for tall, it means that this capital T is the genotype. It is how the genes are present. And tall is the phenotype, because the thing that you can see and observe, that is the phenotype, and the thing that you cannot see is the genotype. The genetic makeup, you cannot see anyone's gene, right? Now if someone is tall, you cannot find out whether he or she or the plant is homozygous dominant or whether it is heterozygous, isn't it? Because we can't see anyone's genes. So the genes of the organism, the way the genes are present, they are the genotype, and the way the external observable character is there, then that is the phenotype. Clear? Now, we are talking continuously about dominant, recessive, and what it is. Now, dominant character. Now, dominant character is that character that will express itself always. So, the character expressing itself is a dominant character, and the recessive character is the character that will not express itself when a dominant character is present. For example, if capital T, small t is present, do you think the plant will be tall or dwarf?
Tall.
Yes, it will be tall, because this dominant trait will mask the recessive trait. But always remember that the recessive character can only and only express itself in a homozygous recessive condition. Because now in this condition, there is no one to mask it, right? So homozygous condition or homozygous recessive condition is when the recessive character will express itself. Any doubts up until now, students? All right. Now you people can note it down. Also, while you're noting it down, first you read it. Okay, first you read what's written on the screen, understand it, and then note it down. Also, while noting down, if you have any problem, you ask. Okay.
Ma'am, it's Maghrib time over here, so can I take a salah break?
Sure, sure, sure.
Hello, ma'am.
Hello, how are you? Good?
Yes, ma'am.
Okay. Students, those who have not noted it down, you people can note it down till the other students are joining, and then we'll continue the chapter. Okay. Done, students? Shall I scroll? Marzi, are you done noting it down?
I'm almost done. I'm at the blue part in the
Okay, okay.
then
Yeah, yeah. What was the portion for the previous test that you people have had here?
Uh, the last previous test we had, the latest test we had full reproductive health and all of reproductive organs, sexual reproductive organs. Second chapter.
Second and third chapter.
Yes, ma'am.
Okay. Done. Shall I scroll down, Marziya? Also, students, did you have any difficulty in solving the questions in the previous class from the topics of the previous class? No difficulties? Everything was clear, right? Okay.
Yes, ma'am.
Okay, good. Let's just wait for others to join and then I'll start from the new topic. Also, you people had notes of reproductive health left, right? Isn't it? There were some points that were left
Yes,
to
ma'am.
note down.
Yes, ma'am.
Okay,
We
today
had
we'll
to
note
focus.
down. Yeah. Today we'll note down that also. Let us wait for all the students to join. Then I'll share it. Okay, students, are you able to see the screen?
Yes, ma'am.
Okay. Now, from where did you people had to note down? Side effects of contraceptive methods were done. So you people had noted this down? No. Done. All right. Okay. From here, I guess. Okay. Now you people can note down. So this is, now you can switch off your video, note down comfortably, and tell me once you're done so that I'll scroll the screen for you.
Done, ma'am.
Everyone's done?
1 minute, ma'am.
No,
Okay.
ma'am.
Okay, I'm waiting.
Done, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Yes, ma'am.
Done, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Yes, ma'am.
Ma'am, for gift, we have to write the whole definition.
For which one?
For gift,
Gift.
yes, you have to write the whole definition.
Yes, yes, yes. You have to explain the condition, like why is it happening? Like why the gamete must be transferred. There must be a reason, right? Because the female, she cannot produce the gamete, or even if she can produce, she cannot provide, like, no, she cannot produce one, but is capable of providing a proper environment, meaning her ovaries are not functioning well, but her uterus is functioning well. So she just needs the gamete. So you have to explain the, you can say, the condition why this is being done. Okay.
Okay, ma'am, thank you.
Everyone's done.
No, ma'am.
Okay, okay. Done, students?
No, ma'am.
Okay.
Done, ma'am.
Done, ma'am.
Everyone's done?
Yes.
Done, ma'am.
Everyone's done?
1 minute, ma'am. Done, ma'am. Done, ma'am.
Everyone's done?
Ma'am,
Yes.
genital herpes is also not curable, right?
It's also not curable. Mention that also. Very good. Rest all the sexually transmitted diseases are curable if they are detected at an early stage. Then only. Okay. And as far as the symptoms of these diseases go, you can refer to your NCRT book. That's the exact same thing that I was going to write only. So you people write that line also. Okay. Now coming on to the topic. So did you people have any difficulty in these? Anything that we have discussed in the previous class?
No, ma'am.
Okay. All right. Now everyone switch on your videos. How many of you here take part in certain extracurriculars at school? Like, are you a part of something in school? Certain sport or anything?
Yes.
Okay. Which extracurricular?
Football.
Football. Okay. What about you, Yahya?
Tennis.
Tennis. Okay. What about my girls? Are you people any like taking part in any extracurriculars?
Yes.
Yes. What is it, Khadija?
Badminton.
Badminton. Okay, great. Okay. Inaya, are you back? All right. So we had done till here, right, students? Till the law that Mendel gave, the law of inheritance. Did you people have any difficulty in this? Shall I give you a rundown again?
Yes.
Can you explain law of segregation again?
See, so Mendel, he is known as the father of genetics. We do know that. Now, why so? Because he gave the law of inheritance and he laid down the ground rules or the basis for how inheritance takes place. Now, he gave two laws under the law of inheritance. The first one is law of dominance, and then there is law of segregation. Now, the law of dominance expresses or tells us that in the F1 generation, all the dominant traits will be expressed, while in the F2 generation, both the dominant and the recessive trait will express itself. Then the law of segregation states that two traits that are present in the individual do not get mixed up, meaning traits such as one, you can say genotype is carrying the trait for a dominant character also and a recessive one, but they will not get mixed up. And both of the traits will be recovered in the F2 generation. Now we'll understand it better when we talk about the cross. Now we have to discuss about the first type of cross, that is monohybrid cross. Now, mono means what, students? Mono? In general, mono.
One.
One. Very good. So, mono means one. Now, this explains the inheritance of only one gene. So, in this type of cross, it is the most basic type of cross. And in this, we are going to find out how one gene is inherited in the organism. So, we take one character into consideration for monohybrid cross. Now, you remember what was true breeding? What is the meaning of true breeding? Students, what is the meaning of true breeding? True breeding means self-pollination, right? The plant, yes. The plants or the organisms that do self-pollination, they are known as the true breeding plants. Okay. Also, the true breeding characteristics, if it is present in a dominant, meaning this homozygous dominant condition or homozygous recessive condition, this is the true breeding quality. Now, for this particular experiment, that is the monohybrid cross, Mendel crossed true breeding tall variety and true breeding dwarf variety. Now, see, as far as the questions from this chapter goes, you'll find it a lot of times that they have said true breeding. Whenever there is the mentioning of true breeding, always remember that if it is true breeding tall, so capital T, capital T. True breeding dwarf, small t, small t. Same goes for all the traits. True breeding means it will have homozygous condition. Clear? Now, we are going to cross. Now see, whether it is dominant, whether it is recessive, what are we doing? We are crossing one character only. Like we are taking one character, that is the height of the stem. Now, the tall plant, tall is the phenotype, and capital T, the true breeding is the genotype. Dwarf is the phenotype, and small t is the genotype. Now, during the cross, what happens? This T will first come and combine with the small t. Like the genes from this side will combine with the genes of that side. This is how we do the crossing. Okay. So this T with that. So we'll have capital T and small t. Then this T again with the next small t, and we'll have capital T and small t yet again. Then now this one, this second capital T will combine with the first small t and form capital T small t. And again, that will happen with the next small t, and we'll have capital T small t. So now these organisms, these are the genotypes of the F1 generation. This is the first filial generation. Now, if I were to ask you students, what is the phenotype of the F1 generation plants? What is the phenotype?
Tall.
Tall.
All of them are tall, isn't it? Now, if I ask you what is the genotype, so you would say they are heterozygous, isn't it? Because they have one dominant character and they have one recessive character. Now, answer one thing, students. Why none of the organism is short? Or there we can see small t everywhere, but why isn't it dwarf?
Because the
Dominant.
dominant trait is capital
Because
T.
of the law of dominance.
Exactly, because of the law of dominance means that the dominant trait is not letting the recessive trait to show itself or to express itself. Very good. All right. Now, here, what we do after this process is done. Now, see, again, giving you a very hack for all your questions is that in many of the questions, they'll ask you to find out the F1 generation. Or in many questions, they have given the F1 generation and then they ask you to find out the F2 generation. So the method of finding the F2 generation is to do selfing. So you take any of them and two of them, actually any two of them, and then you cross them together. So we take now capital T small t, cross it with capital T small t. Now what we do, capital T, when it comes with capital T, it is capital T, capital T. Then this capital T, when it binds with the small t, so we have one capital T and small t. Then the small t will bind, I'm saying bind, but it's not bind. It's actually making a cross with, okay? So this small t will cross with the capital T, but always remember, when any recessive character combines with a dominant character, but still the dominant will be written first. So that is why even if this small t is combining with the capital T, I've written capital T first and then I've written the small t. And then the small t will combine with the small t and we have small t, small t. Now in the F2 generation, tell me the phenotypes. What is the phenotype of this first one?
Tall.
It is tall. Everyone agrees?
Yes, ma'am.
Okay, what is
Yes,
the
ma'am.
phenotype of the second one?
Tall.
Everyone
Tall.
agrees? Third one?
Tall.
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 And the fourth one?
Short.
Short.
Dwarf.
Dwarf, right? Now you see that out of all the members of the F2 generation, 3 of them are tall and 1 of them is dwarf, right? So remember the segregation, the law of segregation or the law of dominance also. It said that both the characters will express in the F2 and only the dominant one will express in the F1. Okay, so now with this, with the F2 generation, we find out the phenotypic ratio, the ratio of the phenotype. So 3:1. Now, do you people understand how the ratio is coming out to be 3:1?
Yes.
Yes.
How?
Because in three of the genotypes, T is the
Is
dominant
what-
one. So
No, I'm talking about the phenotypic ratio. How is the phenotypic ratio coming out to be 3:1? Explain
because
this.
three of them are gonna
Are
be
tall
tall
men,
and one is
and the
dwarf.
one is dwarf.
Yes, 3 of them are tall and 1 is dwarf. So the ratio comes out to be 3:1. And the genotypic ratio comes out to be 1:2:1. Now tell me, is this justified? Is it
Yes,
right or
ma'am.
wrong?
Yes,
It's
it's
right.
correct.
Because one of them is homozygous dominant, 2 of them are heterozygous, and 1 is homozygous recessive. So this is how we find out the phenotypic ratio of a monohybrid cross and a genotypic ratio of the monohybrid cross. But most important is the phenotypic ratio. No one asks you the genotypic ratio. Mostly the phenotypic ratios are asked. So you now understand how to make the crossing. Okay, now do one thing. You people do monohybrid cross. This is for all of you. Monohybrid cross of true breeding axial flower with true breeding terminal flower. And tell me, what do you get? Everyone, do this.
Done, ma'am.
Okay, everyone's done. First, tell me the genotype of the true breeding axial flower.
A.
What AA? What kind of AA?
Since it was axial, I took capital A, capital A.
Because axial is a dominant trait. And axial and terminal are the traits of which character? What is the name of the character of this trait?
Flower position.
Flower position. Very good. All right. So what is the genotype of the true breeding axial flower, students?
Capital A, capital A.
And then what is the genotype of true breeding terminal flower?
Small t, small t.
No. No. Remember, as I told you, to not complicate the process, the genotype of the recessive one, you should take the small letter of that you've already taken. For example, you took capital A for axial. Now for terminal, you do not have to take T. Take small a, small a. Okay? This is one mistake you shouldn't do, because later on it will get very confusing. So that is why you take capital A for true breeding dominant and small a for true breeding recessive. All right. Now you tell me, what are the genotypes of the F1 generation?
Capital A, small a,
Capital A, small a, capital A, small
capital
a,
A, small a,
capital A, small a, capital A, small
Very good. Now I am asking you to find out the F2 generation. What are you going to do?
self a,
a.
two of the
True
capital A,
breeding.
small a,
For F2, yeah.
breeding.
For F2 generation, you have to do selfing, isn't it? Selfing means
Yes.
cross pollination in between either of the member of the F1 generation, right? So now we take this and we cross it with this, isn't it? Now what do we get in the F2 generation, students?
Capital A, capital A, capital A, small a, capital A, small a, small a, small a.
Right? And then again, what is the phenotypic ratio?
3:1.
3:1.
3:1. Was everyone able to do this?
Yes.
Yes.
I'm right. 3:1. Everyone was able to do it? Every single person here? All right. Okay. Now, this was monohybrid cross. Now, does anyone have any doubt here? All right. Now, there's another thing, that is test cross. Now, let us understand what test cross is. Now, when an organism showing a dominant phenotype is crossed with the recessive parent instead of self-pollination, this is test cross. For example, instead of crossing you can say, see here, like for F2 generation, we did this thing, selfing, but for selfing, we took capital A, small a and capital A, small a. But now instead of that, you take recessive parent, meaning capital A, small a with small a, small a. This is known as a test cross. When we cross with the recessive parent, see here, when an organism showing a dominant phenotype, remember this is not dominant genotype, it is dominant phenotype, is crossed with the recessive parent instead of self-pollination. This is known as test cross. Now this is such a bizarre thing, isn't it? One might think, like, why are we doing this? And the reason why we are doing it so as to test or determine the genotype of the organism. When we want to find out what is the genotype, then we do a test cross. Is it clear to everyone? All right, now you people can note down. Then we'll move forward. You can switch off your cameras and note down. Done, students?
Yes, ma'am.
Yes, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Yes,
Now this cross, you made it on your own, or do you need to see it?
We can make it on our own.
Okay, very good. Also, has everyone here understood like how to make the crosses? How to do the monohybrid cross? Everyone, like, is it clear to every single person here?
Yes, ma'am.
What about Ikra? What about Umar? What about Amina?
yes, ma'am.
Okay.
Done.
Everyone's done?
One minute.
Okay.
Done.
All right. Okay. Now let us move forward and talk about the exceptions of Mendelian principles or Mendelian law. Now, Mendel gave the law of dominance and the law of segregation, right? But yet again, as I've always told you, that biology is in itself a science of exception. So we have a lot of exceptions everywhere. And with that being said, let us see what are some of those kind of crosses or some of those kind of inheritance that does not follow Mendel's law. Most of the inheritance do, but some does not, and these are the ones. The first is incomplete dominance. Incomplete dominance. Now, what is it? Example is the flower color of snapdragon or the dog flower. Okay, snapdragon is the name of a flower, and enter hynum is the this thing. It is the scientific name. Now, as far as the inheritance in this flower goes, now the flower color here, the dominant color is the red color in this flower, and the recessive color is the white one. So, when the organism undergoes the process of crossing, so from this, the gamete comes out to be R, and from here, the gamete comes out to be small r. And when they are crossed together, in the F1 generation, all the organisms are capital R and small r. Now, one would think that, okay, this is red. But the case is that in this case, in the F1 generation, all the organisms are pink in color. All the flowers are pink colored flowers. Okay, which is very beneficial to me, but it is something out of the ordinary, right? Why? Because normally we were seeing that in the F1 generation, only the dominant trait was happening. But here, neither did the dominant trait come along, nor did the recessive one. Here is the intermediate. You might if anyone here does painting and stuff, you might have noticed that when you mix white and red together, you get a beautiful shade of pink. So that pink is what comes here. So it is like a middle, you can say intermediate kind of phenotype that is found. Okay, so this is,
So it doesn't follow the Mendelian laws.
no, it doesn't. That is why it comes as an exception. This is happening normally. It is a natural phenomenon. And that is why it comes under the example for exception for Mendel's law. So here, intermediate color is seen in F1 generation. Now, when we do selfing, so we get one dominant, homozygous dominant red, then two of them that we suppose them to be heterozygous but dominant, but no. We have two of them being intermediate colors, and then one is recessive, one is dominant, and two of them are intermediate colors. So now here the phenotypic ratio Normally, the phenotypic ratio in all the monohybrid crosses will come out to be 3:1. But here it's coming out to be 1:2:1. Same goes for the genotypic ratio, 1:2:1. So this is one kind of a cross wherein the phenotype and the genotype ratio is same. Everyone has understood this thing? Also, the most important thing here is to remember the examples. Like in which organism does this phenomena of incomplete dominance taking place? Now, incomplete dominance is when an intermediate character is being shown in the F1 generation. Clear to everyone? Now, the next
Yes, ma'am.
condition is of codominance. Now, both the alleles exist or express themselves equally. And the best example is ABO blood grouping. We know that there is A blood group, there is blood group B and O. But we also know that there is a blood group that is known as AB. How many of you are AB positive or negative here? Is there anyone with AB blood group? Also, how many of you are A? None of you are A blood group
No.
holders. None of you. How is that possible? I have so many students in the class and none of you are A positive or negative. Okay, how many of you are B group blood group holders?
Me.
That's me.
Okay. And then who is O? Okay. Then I am the only one who's A. Here I am the only one who's A positive. Okay. And who is O negative here? Is there anyone who's O negative? No, Yaya, you are O positive or negative?
I'm open.
Negative. And
Negative.
okay, Tamim, you are O positive. Oh,
Yes.
Yaya, if you ever go to donate your blood, people will, they will give you so much of bribes. Like they'll be like, I will give you anything to eat. You name it and you'll have it. Please donate some blood because O negative is such a rare blood group. All right. Okay. So this O blood group system, it shows codominance. We know that A blood group A exists as A, right? B exists as B, and even O exists as O. But then what is this AB? And no one is AB here. So AB is when A is also being expressed and B is also being expressed, which is again a very peculiar thing because when we talked about monohybrid cross, we said that only the dominant will express and the recessive will get masked, or in a homozygous recessive condition, the recessive will express. But here, both of them are being expressed. Now, see, this is how we do the crossing of AB O blood grouping. See, the A blood group, it has the genotype of the A blood group is IA and IA. B is IB. AB blood group is IA and IB. See, this is the IG. Okay, IG. The IG, now, if you people might remember from body fluids and circulation, the blood group system, you might know that the whole entire blood grouping in our body is because of the presence or absence of antigen that makes the antibodies, right? So here, A blood group has the A antigen and it makes the antibodies for B. B blood group has antigen and it makes antibodies for A. Then AB blood group has antigen for A and antigen for B also, but it makes no antibodies. And then O, it has, you can say no antigens, but it makes antibodies, A and B both. So this is the reason why, since the blood group AB does not make any antibody, so the blood group AB can take blood from any blood group. It can take from A, it can take from B, it can take from O. And since O does not have any antigen, but it has the antibody, so it cannot take the blood group from any other. It has to take the blood group from its own type. So with that being said, AB is known as universal recipient. Universal recipient. And O is known as universal donor, right? Okay. Now we are going to see. Now see, out of these two, like IA or anyone, so one comes from the maternal side and one comes from the paternal side, the father side, because the two genes, they are one from the maternal side and one from the paternal. Now, in this chapter, many a times you'll get the question as to how, okay, one more thing. They'll ask you like, the mother is this blood group, the father is this blood group, find the blood group of the child. They'll ask you like this. Okay. Another thing to remember, when IA, wait, I'll not mess this thing up. Wait, I'll write it here.
What is IA?
I is I gene. It is the name, just like how A was the gene or capital T was the gene for tall.
Yeah.
Same way, I is the gene that is used for blood grouping.
Oh.
Okay, and if this gene carries the A, then it is the gene that is coding for A blood group. Now, A blood group can be seen in these two conditions. Okay, this is the homozygous dominant condition, and this is the heterozygous condition, wherein this is the dominant and this is the recessive. So even though small I gene is there, it will still be blood group A. Same goes for B, but the O again, since it is small I, so it will express itself only in which condition, students? Give me the right name. O will express itself only in one condition. What is the name of that condition?
Homozygous recessive.
Homozygous recessive condition, right? Now, but the AB, when I gene is, it is carrying the A antigen, and I gene, another I gene is carrying the B antigen, so it will express as AB, because both of them will get expressed. Okay. Now, this is the chart that looks confusing, but it is not. See this. As I told you that out of the blood groups, one will be contributed by one parent and another will be contributed by another parent. Now, if we are talking about the blood group A, right? So, IA from one parent and IA from another parent. It will give us 2IA, which is the homozygous dominant condition. And the antigen present will be A antigen, and the blood type is A blood group, right? And the antibody will be present against B, because A will not form antibodies against itself, right? It will form against the foreign thing. So B. Then goes for the B blood group or AB blood group. Now, if A is being contributed by one parent and IB is being contributed by another parent, so IA, IB, and both the types of antigens will be there, and the blood group will become AB, but they will have no antibodies. Because what will they make antibodies for? If it will make antibodies for A, then also it will get destroyed. If it will make antibodies for B, then also it will get destroyed. So it has no antibodies. Clear? Now, IA and small I. So as I told you, the small I will get suppressed. It will not express itself. So now the blood group is A. Again, the antigen is A. So it will make the antibody against B. Clear? Everyone is getting this, now? I hope I'm not talking to myself. Everyone is getting it? All right.
Yes.
Now, okay, very good. Now talking about B. For example, if one parent gave IB and another parent gave IA, then also it will be IAIB, and A and B antigens will be present and the blood group will yet again be AB, right? And again, no antibodies. Then if IB condition, like one parent contributes IB, another contributes IB, then B blood group is there and it will make antibody for A, right? It will attack on the external one. Same goes for the recessive condition. It will make antibodies for A. Now coming on to the O blood group. It has two recessive genes. Okay, recessive gene from one parent and another parent becomes small I, and it has no antigen present, but it will make antibodies for A and for B. So as soon as any external blood that may be transfused and it is A blood group, so it will start attacking, which is a very serious condition. But you people understood this? This whole thing is clear to everyone? How the crossing is done? And tell me this thing, students, did you people understand that why are we calling this as codominance? Because of what? Because this chapter, like this topic is not about blood group. It's about codominance, right? I went into zoology. But coming back to genetics, tell me, did you people understand why this is codominance? How many of you did understand this? No one.
No,
See,
I understood this, but I don't know how it is related to codon.
that is what I'm asking. See, the reason why it is known as codependence, you understand? Co means together, right? Cohabitation. Remember, infertility instead of having cohabitation. Co means together. So here, when A is also expressed and B is also expressed, and they are expressed together, and they are not separately, and no one is masking anything, then because of this AB blood group, this is the example of codominance. Meaning the blood grouping system in humans is a great example of codominance. Now, is it clear to everyone?
Only
Yes,
blood group A.
Only blood group AB. No, see, the ABO blood grouping, the whole system, how it works, it shows codominance. But more particularly, which one? So AB blood group. You are right. Okay. Everyone has understood this? Sure,
yes,
100%. So now in the next class, we'll do how to make the crosses. And I'll give you conditions and situations. And in this chapter, the more you can say practice questions, the better you get at. I'll bring, like, once I've explained you the maximum amount of thing in this chapter, I'll bring thousands of questions for you to practice. You'll get annoyed of me, I'm sure, but I don't care because this whole thing will get inside your brain perfectly and I want that for you. So this thing is clear to students? Does anyone here has
yes,
any doubts up until now? All right, very good. I'm very happy to see that. Also, your test is coming up. Okay, the weekly test that is supposed to come, it's coming up. So I was thinking, since we have not done much in this particular chapter, right? So I was thinking giving till monohybrid cross. Okay, not till here, till monohybrid cross. So this chapter till monohybrid cross and reproductive health, sort of like a division. Okay, so these two chapters will come in your test. I'm also writing it down because right now only I've decided. This is clear to everyone, right?
Yes, ma'am. Today I had my assessment.
How did it go?
It was nice.
It was nice.
But
Why is it not convincing enough?
no, the MCQs were like, I don't know.
They were tough.
No, they asked like questions which were not there in the textbook. Two of them.
We can discuss them. Like from which chapter?
Both chapters, human reproduction and reproductive health.
Okay. Okay, we'll discuss it also. And one more thing, students, like I wanted to ask you, like I was very curious, like after studying here, are you people like being like acing in your class? You're like, we already know the topic, we already know the concept. Is everyone like this? Or is it just in my imagination?
In my school, they're ahead of us.
Yes,
So,
ma'am. In my school also
Okay.
they're ahead of us. They're actually here only. They're teaching the blood.
In this chapter.
Yeah. Today I
Okay.
think we did something polygenic. Yeah, polygenic.
Polygenic inheritance. Okay.
Yeah.
That is okay, okay. Okay, you people are enjoying genetics?
yeah.
Is it better than reproductive biology?
Yes.
yes,
Everyone agrees? I'm getting big yeses. Okay. All right. Very good to hear that. Anyway, I hated it when it was my time to study it. But okay. So till now, does anyone have any doubts? No
No,
doubts?
ma'am.
All right. Very good. Okay, then students, I'll meet you in the next class. We'll note it down first in the next class and then we'll continue with it. We'll talk about the dihybrid cross and everything. Okay. Also, did I ask you people to remind me something in today's class? I feel like I did ask you something. I don't know. I always forget.